All right, so the next thing we're going to take a look at now that we've done a little bit on the history of DNA and the discovery of DNA is taking a look at the actual structure of a DNA molecule in more detail. So one of the things to think about when we are talking about the structure of DNA is it is double-stranded, okay, so that's really, really important, so composed of 2 strands, and then you'll notice that it says 2 strands of individual nucleotides. So we're going to revisit a little bit about what nucleotides are, but essentially if I go to our graphic here, this would be a nucleotide, okay, so kind of you can see there's 3 parts here. There's this circular atom with a P in there, that stands for phosphorus, and then we have this pentagonal shaped thing, that's a deoxyribose sugar, a pentose sugar, and then we have our nitrogenous base here, okay? So each one of those is considered a nucleotide or the building block of DNA. The other thing, the other takeaway on this particular slide is that the DNA strands are anti-parallel to one another. So when we think of parallel, we think of like train tracks running in the same direction, right? But a DNA strand is what we call anti-parallel to one another. So if you look at the sketch here of the hands, this is from a very famous artist named Escher, and these are Escher's hands. And so you can see how the 2 hands are like this, okay? They're anti-parallel to one another. That's what you always want to think about with DNA strands. So they don't run parallel to each other like a set of train tracks. They're anti-parallel to one another. And indeed, if you look at this particular graphic, you can see that on one side of the DNA strand, we have something called 5' end, and then down here 3' end. And then if we look on the other side, we have the opposite, right? So running anti-parallel, we have the 5' end and then the 3' end on the top, okay? So you can see that that's what we mean when we say anti-parallel, is they are running this way to each other, just like Escher's hands there. All right, so when we talk about the nucleotide, just like we did on the previous slide, the nucleotide is consists of 3 distinct things. So because we're talking about DNA, which is deoxyribonucleic acid, the sugar is deoxyribose. And this is a pentose sugar, and remember what that means. Pentose sugar means that it's a 5-carbon sugar. So don't think of it as, oh, it's a pentagon, so that means that it's a pentose sugar. It actually is the number of carbons, okay? And then we have that phosphate molecule, okay, so that's the second part. And then we have the nitrogenous base. And of course, remember for DNA, the nitrogenous bases consist of the adenine, the guanine, the cytosine, or the thymine, okay? So if we look at our graphic down at the bottom here, you can see that this is a nucleotide for RNA, and the other one is a nucleotide for DNA. So when we look at DNA, so our nucleotide here, we have our phosphate, okay, group, we have our deoxyribose sugar, which is a pentose sugar, and then we have our nitrogenous bases. Now, one of the things you should notice is different though between DNA and RNA is that the nucleotides in RNA have that phosphate group, which is the same, but you'll notice that instead of the deoxyribose sugar, we have ribose sugar. So it's still a pentose sugar, it's still a 5-carbon sugar, but it's ribose instead of deoxyribose. The other thing that you should notice with the RNA nucleotide compared to the DNA nucleotide is we have uracil here instead of thymine, okay? So in RNA, the nitrogenous base uracil replaces the thymine that we would find in the DNA molecule, okay? So important takeaway here is when we talk about the building block of DNA or building blocks of DNA, it is the nucleotide which consists of those 3 parts, the sugar, the phosphate, and the nitrogenous base. Now, remember when we talk about the building blocks, that's what we call the monomer. So that word monomer is in our program of study. So you might remember back when we did Bio 20 that when we talked about monomers of carbohydrates, for example, that would be like a monosaccharide, right? And when we talked about the monomers of proteins, that would be like an amino acid, right? So it's the smallest unit of that particular macromolecule. So for DNA, because DNA is a macromolecule as well, the monomer is the nucleotide, which consists of those 3 different parts. Now, the other thing that you'll notice is that we have these base pairs matching up with each other. So you'll notice that you always have a C with a G, and then you always have an A with a T. So those are the base pairing rules, which we'll talk about a little bit more in the next couple of slides, but those are base pair rules. So you can never match the other way. So you can never put a T with a G or an A with a C. It's always C and G and A and T. Okay, so that's one thing that's very important. And then of course in the RNA, the uracil will replace the thymine, right? The other thing is you'll notice that these base pairs all have this kind of yellow kind of structure in between there, and what that is a hydrogen bond. So in between those base pairs, the rungs of those ladders are held together by hydrogen bonds, okay? So double helix, so we take that ladder and we twist it right into that double helix, and just remember the nucleotide composition. The other thing to remember too is that when we talk about the sides of the ladder, okay, that's what we call our sugar phosphate backbone. And so that's of course this part here, right? So the phosphate and the deoxyribose sugar on both sides of the DNA molecule make up the sides of that ladder. And the other thing that's important to remember with respect to that is that sugar phosphate backbone is the same for all living things. So it doesn't matter if you're a snail, if you're a human, a chimpanzee, if you're an amoeba, doesn't matter what kind of living thing you are, the sugar phosphate backbone is identical for all living things. What makes us different is our sequences of base pairs, okay? So how are these base pairs matched up together? It's the same for all of us in that we all have the adenine matching with the thymine, and we have the cytosine matching with the guanine. That's the same for all living things, still the same nitrogenous bases, but the order of those nitrogenous bases on and how they match up on our genes is different, and that's what creates the different proteins and therefore different characteristics of different living things. So if we take a look at our next little slide here, as we said, there's those 4 nitrogenous bases, and we can break those down further into pyrimidines, which are single-ringed nitrogenous bases, and purines, which are double-ringed nitrogenous bases. And this part is actually in your data booklet, okay? So you don't really have to memorize it because it does tell you in your data booklet which ones are the pyrimidines and which ones are the purines, okay? So just taking a look at that graphically in terms of the chemical structure, so you can see that the thymine and the cytosine, those two are single ringed, and then you can see with the adenine and the guanine that those two have the double ring structure, okay? So the pyrimidines are the single rings and the purines are the double rings. And one thing to always remember too is you're always going to have a pyrimidine bonding with a purine. So A will always bond with T and C will always bond with G. And this is what are called the complementary base pairs. They complement each other and they attach to each other in the formation of a DNA molecule. One really easy way to remember this is just to remember that toothpaste name Colgate. This is just one way I've always remembered which of the base pairs actually go together. And so if you just kind of follow reading the name Colgate from left to right, C and G are the first 2 nitrogenous base letters, so C and G always go together, and then A and T are closest to each other, so those are the 2 that will bond together as well. So C and G and A and T. So just remember that, Colgate, and then you'll never go wrong with which base pairs actually go with each other. So moving up to onto our next slide, we already talked a little bit already in the history of the DNA video about the contributions of the different individuals. And so we're just going to summarize again the Watson and Crick model. And remember, they built off the work of Franklin specifically in terms of building their model for actual DNA structure. And it is still the same conceptual structure that is used today. So remember, we call that molecule a double helix. And the 2 nitrogenous bases are joined together by weak hydrogen bonds, okay, so they can be broken with particular enzymes when the DNA actually needs to replicate. And remember, we always have the purines and the pyrimidines binding to each other. So again, that rule of the C bonding with the G and the A bonding with the T, right? So always, always, always. And remember, the sequence of base pairs along the gene is what determines our genetic code. So while all living things have that same sugar phosphate backbone and we all have the same nitrogenous bases, all living things have the G, the C, the A, the T in our DNA, it's the sequence of those combinations that actually determines our genetic code and creates those differences from one species to the next. So another thing that we do have to remember with the bonding is we already talked about the fact that there's hydrogen bonds, weak hydrogen bonds that keep the double helix intact, especially when it's twisting into that double helix structure, and keeps those base pairs attached to one another. But one thing that we do have to notice is that it's different in terms of the number of hydrogen bonds we actually have. So you'll notice when we have the A and the T together, we have 2 hydrogen bonds, but when we have the G and the C together, we actually have 3 hydrogen bonds. And so just like our Colgate scenario, we can kind of remember this by going GIC3. Okay, so the letter G, of course, standing for the guanine. And so if you just remember GIC3, then that will help you remember that the cytosine and the guanine have 3 hydrogen bonds, and the adenine and the thymine have just the 2. So our last kind of thing for today in terms of the DNA general structure, and again, this is general structure, we'll get into more detail when we do the DNA replication, is the work of Chargaff. And we kind of talked about Chargaff a little bit when we said that he's the one that kind of figured out percentages of base pairs in DNA. So in 1950, Chargaff analyzed the base composition of DNA in a number of different organisms, and then he compared them and analyzed them to one another. And what he ended up coming up with is what we still use today to determine which base pairs go together. And so Chargaff's rule is as follows, and this is in our program of studies. It is a very simple type of calculation question. So basically, because of the bonding nature, so how C always bonds with T and A always binds with G, your number of pyrimidines is equal to your number of purines, okay? So what does that mean in an actual practical sense? Well, what it means is the percentage or number of your adenines is going to be equal to the percentage or number of thymines, and the percentage or number of cytosines is going to be equal to your percentage or number of guanines, okay? So sometimes kids are like, what are you talking about? Well, remember, that doesn't mean that A plus T equals C plus G. It means something different. So let's take a look at an example. Okay, so how does this look in a test situation? Typically speaking, what will happen, I'm just going to turn off the camera here quick so we can oh, it's not going to let me do that because I was going to say so you can actually see what's going on here, but while we're recording, it won't let me do that. Okay, that's okay. So 15, let's say that you get a question that says a sample of DNA, okay, has 15% thymine. Okay, so let's say that's what you're given as a question, and then it says calculate the percentages of the other nitrogenous bases. So you're given thymine, and then you're asked to calculate how many adenine, how many cytosine, how many guanine. Okay, so the first thing to remember with Chargaff's rules is remember that your percentage of T's has to be equal to your percentage of A's. So therefore, if thymine is equal to 15% of that particular DNA sample, then adenine also has to be equal to 15% of that particular sample. And then, of course, just like is typical with percentages, we can only go up to 100%. So then your next step after that would be very simply to say, okay, well, my adenine and my thymine together, that adds up to 30% total of my DNA sample. And then obviously now I have 70% left. And because your number of cytosines or your percentage of your cytosine has to be equal to the percentage of your guanine, well, that's where the 35 comes from. So 30% was thymine and adenine, and then 35% for each of cytosine and guanine, because then that gives us our total of 100%. And we'll do some practice with that in terms of worksheets, etc. So that will become something that's quite easy for you, but it is a required piece.